Thank you, everybody, for joining us today. Really excited to have Rachel Smith with us as one of our neuroengineering faculty candidates. So Rachel grew up just up the road in Chattanooga, went to University of Tennessee for her undergraduate, and then did her graduate work out at Irvine. And she's now at Johns Hopkins doing really exciting work that you'll hear about today in her talk, Dynamical Network Models of the Epileptic Brain to Improve Surgical Outcomes. And she's also been doing some really interesting teaching as part of her postdoc at Morgan State, which is what we were chatting a little bit about pandemic teaching, if some of you caught that as you were coming in. Rachel, it's great to have you with us, and we're looking forward to your talk.
Awesome. Thank you, Dean Holmes. Appreciate it. Y'all, I'm so excited to be here. Thank you so much for having me. As mentioned, my name is Rachel Smith. I'm a postdoc in the Neuromedical Control Systems Lab headed by Sri Sarma at Johns Hopkins University. And we're also part of the Institute for Computational Medicine there. And I'm excited to tell you about one of my postdoc projects that I have been doing for the past couple of years, using some dynamical network modeling to hopefully improve surgical outcomes in epilepsy patients. So let me make sure this is working here. Whoop. There we go. So first we'll talk about what epilepsy is. So epilepsy is a neurological disease that is defined by recurrent and unprovoked seizures. And so a seizure is this sudden electrical activity in the brain that changes. So if you look at this bottom right-hand figure, you can see that there's a lot of squiggly lines. So each one of those lines is recording from a scalp EEG electrode on a pediatric patient. And so it's measuring the brain voltage over time. And as you can see in this white box, there you can see the activity kind of changes, right? It goes from this low amplitude, kind of mixed frequency, to being a lot more high amplitude and kind of more of this rhythmic pattern. And this is an absent seizure that happens in kids. And so these seizures can be associated with like a loss of awareness, a loss of consciousness, or loss of motor function. So pretty debilitating for people's quality of life. So there's about 30% of people who have epilepsy that have what's called medically refractory epilepsy. So first line treatment for epilepsy patients is they're going to be given anti-epileptic drugs. But for about 30% of these patients, the drugs are insufficient to control their seizures. And so surgery can be a therapy for these people if they have what's called focal MREs or focal epilepsy, which means that the seizures are kind of generating from one part of the brain and then spreading to the rest of the brain, as opposed to a generalized epilepsy where it kind of looks like the seizures are kind of starting all over the brain at the same time, right? So if clinicians can localize where the seizures are coming from, right, then they can go in surgically and take out that part of the brain, or they can ablate it, or you can use electrical stimulation and stimulate it. But the idea is that if you can remove this part of the brain or stimulate, modulate it in some way, then these patients can be seizure-free after surgery, right? So first we have to localize where the seizures are coming from, right? So there's a couple ways that clinicians do this right now, right? So there's typically you have to get into, so for these patients that are not really easy cases, if they'll do a lot of non-invasive imaging and they'll decide, okay, I think we need to go with implanted electrodes to kind of see where the seizure is coming from, kind of in more of these deep brain structures. So what they'll do is they can, if a patient has an ECOG grid placed, they can sometimes take off part of the skull, do a craniotomy, and place these ECOG grids across the cortex. And so they can measure kind of these more neocortical activities. And then you can also have what's called stereo electroencephalography or stereo EEG, and they'll drill these electrodes into the brain through these little burr holes, and that can help them measure some of these deeper brain structures that you wouldn't be able to really see with some of the ECOG grids. And then the patients will sit in the hospital for anywhere from a couple days to record seizures. But sometimes if they don't have enough seizures, they can sit in the epilepsy monitoring unit called EMU. They can sit there for weeks to have enough passive seizures so that clinicians can visually look at the data and see, they'll look for features of the data that are kind of indicative of where the seizures are starting. So those can be kind of high frequency oscillations, those can be paroxysmal shifts in the data or like a beta buzz, but they'll look for these features in different channels of the EEG to decide where the seizures are coming from, right? So this is a pretty invasive process, it's a long process, it's an expensive process, and unfortunately, about half of these patients that come into surgery will come out of surgery still having seizures, right? And that can vary for different types of it. If you have a pretty, what they call temporal lobe epilepsy or a mesiotemporal epilepsy, these are pretty easy to treat, and their success rate is about an 80%, right? And especially some of these more, like if the epilepsy is more multifocal, meaning it has different foci all across the brain, or in some of these like harder to reach places, they can be as low as 34%, right? But average, a lot of patients come out of this surgery still having seizures. And we think that there's a way, like, how can we improve this process, right? So there are two things that I think, there are two ways that I want to target this whole process and kind of make this whole process better for epilepsy patients. One, I think, is we want to try to have better ways to localize where the seizures are starting. And then secondly, if there was a way that instead of having the patient wait for 3 weeks for a seizure, right, what if we could stimulate the brain and actively probe the seizure network to expedite this process to where we can have a patient come in, and we can probe this network for a couple days, and then they could go to surgery right after that instead of having to wait so long. Can we expedite this whole intracranial monitoring process? So those are two goals that I have tried to tackle with my current project. So I'll tell you one hypothesis about how we're going to do that, right? And that actually it's best exemplified with a little story. So back in 1997 in Japan, about 700 children went to the hospital for seizure-like events. And it was because they watched a specific Pokemon episode that had a 12 hertz flashing light in one of the scenes. And it caused this, like, what I call a photosensitive epileptic event in all of these kids, right? So basically there's this flashing light, and that caused the synchronization of some of the activities that are happening in their occipital lobe where all your vision is processed. And then it caused them to have this epileptic event. So you have this like synchronization of all this activity, and it propagates to having a seizure, right? So as an electrical engineer, right, I can think about this in a sense of resonance, right? So resonance is this idea that you can have a system, right, that if you push it at exactly the right frequency, you can kind of magnify the output of the oscillations that can come out of your system. So we can think about this as an example of like pushing a child on a swing, right? If we analyze the amplitude of the swing, so how high the kid's gonna go, right, is as a function of how hard or what frequency we push them, right? So if we push them too slow, right, then they're gonna come back to equilibrium with the bottom and they're not gonna go very high, right? And if we push them too fast, they're not going to swing very high because we're pushing them too fast, right? But if we push them at exactly the right frequency, the swinging frequency, or what we're going to call the resonant frequency, the amplitude of their swing can go higher and higher and higher, right? And there's actually a really cool example of this in a real physical system. This is the Tacoma Narrows Bridge. I'm going to turn this sound off. Yeah. So this is a bridge that was built in around the late 1930s, 1940s. And it was built in a way that when there was a really high wind day, and this would happen a lot actually, and they noticed that on these like really windy days, the wind would hit the bridge at exactly this resonant frequency, and it would start to swing like this. And look, I mean, this is like insane, and this is not a cartoon, like this is real life. And actually this bridge collapsed in 1940 because the wind was rocking this bridge so much, because it was hitting exactly its resonant frequency of the system, and it caused this whole bridge to collapse, right? So what we're wondering, my hypothesis is that, hang on, let's go. Okay, there we go. My hypothesis is that what if this theory can apply to all focal epilepsy, right? So we've seen that it can happen in this instance of photosensitive epilepsy, right, where a flashing light can trigger this epileptogenic activity. But what if that's true of epileptogenic regions in general, right? Like, and kind of, you can think about this in the sense of some people look at these different dynamical systems of like in bifurcation theory, right? If you have a certain type of noise that hits the system in the right way, you can push it into more of a seizure-like state, right? But what if there's a specific periodic stimulation that you could enhance the oscillations in a way that would trigger a seizure? So we want to ask the questions, are epileptic brain regions highly resonant? And then can we guide the stimulation of seizures using this resonance hypothesis? So our main hypothesis is that we do think that resonant regions are going to correspond to these seizure onset regions, and that we can trigger seizures when we're stimulating at this resonant frequency. So in the way that we're gonna measure this is we're gonna look at the brain as a system, or the brain as a network, right? So there are tens of billions of neurons in your brain, and they're all interconnected, and they're hierarchically organized so that you can get macro scale dynamics and you can get these micro scale dynamics that all are working together to form your cognition, to form all these processing things that your brain does on a daily basis, right? So we're going to use the fact that your brain is this really cool complex network, and we're going to build and we're going to see how people in general these days look at this network. And that's usually, and one way that people are starting to look at this network is using single pulse electrical stimulation. So the idea behind SPES, or single pulse electrical stimulation, is that you are building what's called an effective connectivity network. An effective connectivity network means that you are seeing a directed pattern, right? So you can actually say the activity in this region is directing the activity of another region, right? So the way that they're doing that is you can stimulate, once you have these electrodes implanted in the brain, right? You can stimulate a pair of electrodes, and you can measure the responses in all of the rest of the brain, right? And from those responses, you can kind of statistically look for these like a high amplitude response. You'd say maybe if by some, typically usually by some z-score that's relative to some pre-stimulus baseline distribution, right? But you can say that if you see a significant connection, that there's an anatomical or functional connection between the region that you stimulated and the region that you're measuring from, right? So on the right, you can see an example from one of the grad students that I work with. He's showing an example of when you stimulate at one electrode, and then he draws a line by and showing the connection strength between different regions by looking at the amplitude of the z-score of the amplitude of the response region. And we have this hypothesis, and in general, this has been shown in SPS literature in general, that when you stimulate or measure from epileptogenic regions, you can usually see a higher amplitude response, meaning that there's this like hyperconnectivity or this hyper excitability of the cortex in epileptogenic regions. So we know that SPES is going to be able to show us that it's going to have some implications for highlighting regions of seizure onset, or these epileptogenic regions. So this is the data, this SPES data is what we're going to be using to build our dynamical network models. So the way that SPES is done, and the way that we've done it in our studies, is that we will stimulate a pair of electrodes, and we're using a 150 microsecond pulse biphasic, right? And we're going to stimulate at half a hertz. So every 2 seconds, we're going to stimulate, and we'll get a response waveform out. And then we can time lock the responses to that stimulus, and then we can get an averaged trial response waveform, right? And so typically what you're seeing on the right is an example of what's called a CSEP, or cortical evoked potential. And then you're seeing this first peak is called an N1 peak, and that's typically thought to represent some of these more like direct anatomical connections, almost, where it's like more local connections of like a U fiber connection in between two regions, that kind of thing. So that's going to be really short. It's like 10 to 50 milliseconds usually. And then the N2 peak is a peak that happens about anywhere from 100 to 500 milliseconds after the stimulus, and it's really thought to represent more of the subcortical dynamics. So and so kind of subcortical driving, so something that's going through the thalamus and coming back out, right? So you have this kind of stereotypical response that we're looking at. We're going to use this average trial response into our dynamical network model. Okay, but jumping back for just a second, let's forget about stimulation for a second, right? We're going to talk about a state space model is a type of dynamical network model that can be really powerful, right? So how we're going to build this state space model is we're going to look at we're going to start with X of t, which is the state of the system. Okay, so what that actually is that's going to be the brain voltage at a specific time point. So, and this is going to be of n by 1 dimension, where you have n number of channels. You're going to pick a time point, and you're going to look at the EEG in that time point, and you're going to say, okay, this is the brain voltage at that time point. And then X of t plus 1 is going to be the new state of the system, or the brain voltage on the next time point, right? And that's going to be modulated by what's called the state transition matrix. Okay, and this is going to be estimated with a least squares estimation. Where the A matrix is an n by n that kind of describes, like, you can think of it as a functional connectivity matrix almost, right? Where you can say the way that this is defined is that, like, if you look at the columns and you say, like, I'm going to look at this node I in the column, right? That's gonna, the way that you can think about each element in the A matrix is gonna say, how does node I affect node J in the next time point, or row J in the next time point, I'm sorry. So basically the A matrix is gonna describe how the state changes from one time point to the next, right? And we can find that these state space models can be really powerful, even in this, so we're staying in the linear regime, but we know the EEG is super nonlinear, right, in most part. But if you look at it as short enough time windows, it can actually match pretty well. So what we've done is in a 500 millisecond time window, I've just measured x of t plus 1 equals ax of t, estimated A using least squares estimation. And then you can see that the red line, which is the model reconstruction of these different models in 500 millisecond time windows, and it matches the EEG, the real EEG data pretty well, right? So we can see that these state space models, even though a really, really simple linear approximation, can capture some really cool dynamics of the EEG, right? Now, when we want to model single pulse electrical stimulation, we're going to need to add a new term. We're going to add an input term here. So U of t is going to represent our input. That's just going to be a step pulse. And then, and that's going to be our, it's a scalar that's representative of our stimulation. And then our input vector is going to be described by B, right, which is an n by 1. And basically B is going to describe how that input is going to affect our state. Remember our state being x of t, right? So when U changes, B is going to help us describe how x of t is going to change, right? And so what's interesting was we can see, okay, well, how well does this capture SPES responses? So this is just kind of some representative examples of one model capturing several different channel responses. So the black line that you can't really see very well, but is the average waveform that's underneath the blue, which is the model reconstruction. And then the purple is showing you a plus or minus standard deviation of all of we usually get about 40 to 50 trials every time that we stimulate a given region. And so, so what you're seeing is the standard deviation of those 50 responses in purple. And so these are showing you some representative examples, but what's cool is that this is true for all of the channels in a given data set, right? So then where the yellow now is showing the 1 plus or minus 1 standard deviation in the from the waveform. So so cool. We have a model that can accurately describe and reconstruct single pulse electrical stimulation data, right? So now what we're going to do is we're going to put this state space model that we're working with into a new form that's going to help us use some properties that are going to help us try to localize where the seizures are coming from. So first we have what we just described, our A and our B in our state evolution equation, and then we're going to change it into what's called a transfer function, which is kind of a different representation of the system. And we're going to add this little scaling factor C here in order to get H, which is a vector of transfer functions that's going to take our input stimulus U and change it into what our multi-output responses to electrical stimulation, right? So now we are set up to build what's called a Bodie plot. So I'll tell you what that is. So so we have all of these responses where we've stimulated the brain, we get all of our responses out, we've built our transfer function, and then what we can output from our transfer function model is what's called a Bodie plot. So a Bodie plot, if you remember, this is very similar to the plot that I was showing you with the pushing the child on a swing, right? So we said that if you push at a certain frequency, so that input periodic stimulation that you're putting into the system, you can reach a certain magnitude of swing, right? So or amplitude of swing. So this is a very similar plot. So where we're showing if I drive this system with a given frequency, what is the maximum output magnitude that I can get out of the system? That's what this is measuring. So so the reason that we can draw this line here, and the reason we can say, okay, how do we know how the system is going to respond when I drive it at 4 hertz? If I drive it at 10 hertz, how am I going to know how the system is going to respond? And it's because we're estimating this SPES response as an impulse response of the system, which kind of has its own set of characteristics that we can derive from getting the impulse response of the system. So yeah, so if we simulate in one area, this is a Bodie plot that we can get. And then we hypothesize that if we're stimulating in what over here is our seizure onset region, we'll get a new Bodie plot, right? A different one. And we can look for these peaks in the Bodie plot. And that's what we're going to assume is this resonance in the system, where there's an increased magnitude in response to a specific frequency, right? So, okay, so now, as I mentioned, our project objectives here are to try to localize the seizure onset better and see if we can do it faster. Can we actively stimulate the seizure network to expedite the intracranial monitoring process? So first, we're going to see, can we localize the seizure onset using these Bode plots? Okay. So to do this, we used a patient data set of 32 patients, and 28 patients were evaluated for surgical outcome after 1 year. And we did all this at Johns Hopkins. And so 20 of our 28 patients were surgical successes, meaning that after surgery, they did not have any seizures anymore, or they had only a few, and it was a significant improvement. And there were 8 that were surgical failures, meaning that they did not do well after surgery, right? And the 4 we're still waiting to hear back from because it's been less than a year. So the way we're doing this, we get our transfer function model, we do SPS, we get our transfer function model, and then we're looking for, we build these Bode plots, and we're looking for different features that may be indicative of where the seizure onset regions are and maybe predict surgical outcome. So some of these features that we pulled out are one called the DC gain, which is kind of what the output of the system is at 0 frequency. And then we can look for the peak gain, the peak frequency, the roll off, the area of the curve, different things. And we also wanted to build a custom metric that would kind of get at this idea of resonance, right? So we needed to know, where do we find these peaks in the Bode plot? How do we characterize them? So I built a custom metric called the PW ratio that looks for these peaks in the Bode plot. So I find where this peak amplitude is, and I find the left local minima associated with that. So we'll call omega left, and we'll call this A min. And then I look for the corresponding right frequency that's on the same level as omega left, and I find omega right, okay? So then I calculate a ratio between the difference in amplitude over the difference in frequency, with this idea that the peakier the plot, that PW ratio is going to be much higher, right? You maximize the difference in A, minimize the difference in omegas. And so in our hypothesis is that the seizure onset regions will have higher PW ratios than non-seizure onset regions. So I'm going to show you just some representative examples of this. So the top patient is a patient that was a surgical success, meaning after surgery, they did not have any seizures anymore. And so we hypothesized that means that that where the clinicians had labeled as a seizure onset zone, when the neurosurgeon took that part out, that was actually the true seizure onset regions, because afterward they didn't have seizures anymore, right? So when we look at these Bode plots, we can see that the regions that were labeled as seizure onset zone had kind of higher DC gains, and they're typically a little peakier, right, than some of these non-seizure onset regions. And if we plot specifically the PW ratios here, we can see that in general they are higher than some of these regions that were not in the seizure onset zone. Whereas for a surgical failure on the bottom, we're seeing that the Bode plots weren't really distinguished between the seizure onset and non-seizure onset regions. In this patient, this patient, we assume that either there were some regions that were still left in that were epileptogenic, or that maybe, we don't really know what happens in surgical failures a lot of times. But basically, it's interesting to see that in the failures we expect that our predictions kind of mismatched what the clinicians were seeing. And so then if we look at this on more of a group level, this is including all the 28 patients that we had gathered. If I use a logistic regression model and I pick features, and basically I just took the difference in the features. So if and the ones we included here were the DC gain and the PW ratio. So I took the difference of the DC gains and PW ratios in the seizure onset regions and subtracted them from the non-seizure onset regions and used that difference as a feature in a logistic regression model. I can predict surgical outcome with an AUC of 0.83 in the Bode plots. And I also compared this to kind of the best metric that we have using these evoked responses, using the CSEP responses. So I used the N1 and N2 Z-score amplitudes also as features to try to predict outcome, and actually did really well. It performed with an AUC of 0.76, but it's interesting, it's not quite as discriminative as the Bode features. And so what we're seeing here on the right are the p-hat values or probability of success. And you can see with the Bode features, they're slightly more discriminative, but still significant using the CSEP features. So overall, we feel like we were able to localize the seizure onset as well as the clinical standard, at least. And what's kind of cool is that we knew that SPES and these evoked responses and the N1 and N2, like those kinds of features are have been shown to be representative of discriminating seizure onset regions and non-seizure onset regions. So the fact that we were able to match that is really cool. But then we're also kind of getting this extra information about kind of what frequencies are most important in the system. So that's so we feel like we accomplished this goal. And so now we're going to move to seeing if we can actively stimulate the seizure network to expedite the intracranial monitoring process. And so the way that we tried to do this is, right, we get our SPES, we build the transfer function model, and we get our Bodhi plot out, right? And if you remember, we are looking for these peaks in the Bodhi plot, and that's associated with a specific frequency that we're calling omega star. So that is the resonant frequency. And so what we believe is that we can stimulate, that it would be more likely that we're more likely to stimulate seizure activity if we stimulate the region at its resonant frequency, right? We're going to probe the network at its resonant frequency, and that's going to magnify the amplitude of the oscillations, right? So we think that this is what might be something to trigger seizure activity. And so this is the first method that we've seen that would be able to guide clinicians if they were trying to induce seizures with stimulation. This would be the first way, that computational way, that they'd be able to guide where and at what frequency to stimulate. And what's cool is that you can do this without needing to capture seizures beforehand. We're not doing this from any seizure data. We're doing this only from the single pulse electrical stimulation data. So this is something that patients could come in, we could do SPES, stimulate for seizures, and then they could go straight to surgery, right? So this is the idea of seeing how we could expedite the process. So let's see if it works, right? So I'm going to show you a couple examples from. So first, we did a retrospective study of, were there any times, and there were, but yeah, so we found all the times that seizures were triggered accidentally in the epilepsy monitoring unit, right? So, and those could happen when they were either one, doing SPES, so was a seizure triggered while they were doing the actual SPES protocol, or if they were doing what's called functional mapping. So oftentimes when patients are going into epilepsy surgery, they need to know where your motor functions are and where your speech processing is and where your language processing is and all those things like that. Those are all regions in your auditory processing, right? Those are reasons that they don't want to take out, because that's going to decrease your quality of life after surgery, right? So what they'll do is they'll go through and they'll kind of stimulate at 50 hertz to try to test some of these functional areas to make sure that they leave those in during surgery. So, but sometimes when they stimulate at 50 hertz, that can trigger a seizure. And so there were a few cases where that actually happened. So we said, okay, if we know that these are the stimulation frequencies and locations that triggered a seizure, let's see if that matches our Bodhi plot predictions, right? So we built Bodhi plots. So what I'm showing here is an example from R002, which is this guy here. And so they stimulated at 50 hertz, and we see that there's a peak at around the 50 hertz region. And so, so it was stimulated during functional mapping, got a seizure in it, and it matches, one of the peaks in our Bodhi plot. What's interesting is for two of these patients that didn't match our Bodhi plots, one of them did not, the seizure that was triggered was not native to that patient. And the other one ended up not being able to go to surgery because the seizure onset regions were too broad. And so we think it's really interesting that it seems like even in the plots that are, even in the patients that our plots really didn't match, there was kind of like reasons behind why it may not have matched. And it may be showing that these Bodhi plots are actually more showing kind of specificity into these seizure onset regions. And then in some of these cases, we tried to prospectively stimulate for seizures. So we would do SPES, I would build the models in like an hour, and then I would tell the clinician, our clinician June, have been working with, I would say, June, I want you to stimulate this region at this frequency and see if we can get a seizure. Okay, so what I'm showing is an example P003, this guy here, where I gave her some of these regions and I said, hey, I want you to stimulate RAH at 18 hertz, and I want you to stimulate LPH at 20 hertz because these are where these really tall peaks in the Bode plot are. And she did, and we actually got a seizure at RAH, and we got an aura at LPH, right? And I specifically wanted to also highlight this, this patient P005. There was a resonant peak at around like 13 hertz, and we actually titrated up. So basically we started out and we said, okay, let's do a stimulation at 1 hertz, 2 hertz, 3 hertz, 4 hertz, all the way up to, and we got a seizure at 13 hertz in one day, right? And then the patient, we stopped stimulating and then came back the next day, and we were able to elicit a seizure again at 15 hertz, right? So I think that it kind of was showing that there is some robustness to some of these measures, that not only we didn't get a seizure when we titrated up, and then we were able to elicit a seizure on 2 different days at the same frequency, which is pretty cool. Oh yeah, and overall we were able to elicit a seizure in 4 out of the 6 patients, or a seizure or an aura in 4 of the patients. Yeah, so we feel like this is going to be a way that we may be able to guide clinicians in the induction of stimulation-induced seizures, which would be kind of a way to expedite this whole intracranial monitoring process if the patients don't have to sit and passively wait in the EMU for 3 weeks to have enough seizures to analyze. So some future directions of where this needs to go in order to actually be translated to the clinic. We need to do a lot more validation study, right? So we've done this in 6 patients, cool. We need to do this on a lot more patients, and there's some specific factors that I think we need to kind of hammer out as we go through this. So one is the fact that different types of epilepsy are going to give different responses to stimulation. So for example, if a patient is lesional, oftentimes the evoked responses that come from regions that are around the lesion will be not as high in amplitude. They'll be kind of reduced because of the scar tissue that's around it. And we actually noticed this in one of our patients. Even though we knew that the seizures were coming from around the lesion, the responses that we were getting were not amplified in any way in the seizure onset. They were actually diminished because it was so close to this scar tissue. So having a group level kind of analysis of different lesion, like MRI positive patients versus MRI negative patients, that's gonna be something that's really important, as well as electrode type. So the fact that you can use ECOG grids and you can use stereo EEG to stimulate and record, but these inherently, they're different sizes, they look at different types of tissue. So this is something that's going to be really important as we kind of control for in a future validation study, as well as electrode location, right? It's been shown that the responses look different when you're a lot closer to white matter tracks than if you're solely in gray matter. So not only in just gray matter and white matter, but also just different regions that are highly functionally connected are going to give you some interesting evoked responses in that way. So having kind of this more anatomical and global electrode location kind of focus is going to be really important for our bigger validation study, as well as just stimulation parameters, right? This is actually something that one of the grad student that I was mentioning earlier in our team at Johns Hopkins have been really active in trying to find what are the optimal stimulation parameters to elicit these evoked potentials. You can change the amplitude of the current, you can change the pulse width, you can change how fast you're doing it. So those are things that are like a standardized protocol across. Usually, I mean, if people are going to be doing SPES, they will probably have a standardized protocol within their center, but those are not going to be standardized across all centers, right? So that's something that we, if we're going to do a large prospective kind of multi-center study, these would be something that we would have to decide. Another interesting future direction that I've just started to look into, I actually just got this data this week, but what if we were able to do any of this non-invasively, right? And specifically magnetoencephalography is a recording technique where it's measuring the magnetic fields of the brain, and it's a modality that is whole brain, it's high spatiotemporal resolution. So we asked the question of, okay, well, this would be the modality where we could look for resonance non-invasively, right? So we have started so one thing that we're hoping to do is to record MEG data, and we can get or we can get MEG data from different places, construct a transfer function model, perform virtual SPES, right? So once you have your model, I can artificially stimulate, virtually stimulate, and then assess Bode plots from there and look for resonance. So this is just showing one example that we got from a patient where we knew that the seizure onset was around this node here. And so when I built one of the transfer function models and I stimulated and looked for resonance. And so this is the seizure onset region is the red. And then I just picked randomly 4 other channels that were non-seizure onset. And you can see that they have different dynamics. I'm not trying to imply anything about this patient, but it's more just that we can virtually stimulate and look for Bode plots, look for resonance in Bode plots in MEG data, which is going to be really exciting. And with that, I'd like to thank you for your time. I'm happy to answer any questions.
All right, thank you so much, Rachel. It's not a huge group. If people want to unmute to ask a question, why don't you just go ahead, and if it gets too chaotic, we can start putting stuff in the chat.
I'd like to ask a question. Great talk, Rachel. My name's Larry. I think we're going to be meeting tomorrow. I'm one of the epileptologists here, and I'm also have a background in engineering as well. So could you explain a little bit more? I mean, everything you said was very, very interesting. I really like this talk. It's one of the best talks I've heard in a while. And
thank you.
Yeah, really related to this stuff. And you did a great job of explaining things. I was a little confused about the MEG data. It seemed like your other data relied on stimulating, like doing kind of a input. Inputting a pulse. How did you get the same thing without being able to stimulate the brain with the MEG?
Right. Yeah. So actually, I think this will be okay. We're gonna go all the way back. So yes, so that's a great question. So what we wanted to do was see if we could virtually stimulate. So the idea, okay. So yeah. So from here, right? So basically, what we've been the way that I was working with the intracranial data, right, is that I would have the data where I knew where the pulse was, and then I knew that then the data changes in response to the pulse. So we get this deterministic output, right? And so I could actually estimate A and B. So I would actually, mathematically, the way I'd actually do this is I estimated A and B simultaneously from the data. Whereas then what I was planning on, what I did in the MEG data is I actually just used this form of the equation for the MEG data, where I said, I knew that there isn't actually any stimulation happening. I just estimate the A matrix, and then I artificially made B a vector of zeros with a 1 indicating where I'm stimulating that electrode, right? And then from that, and then basically I'm just building my transfer function model using A, what it is, B as a vector of zeros with a 1 indicating where I want to stimulate, and then C being an identity scaling factor, and then built my transfer function model to output a Bode plot.
Interesting. Okay.
Yeah.
Well,
so it's very much
cool.
Not do what?
Yeah, I was just say I think this is great. I mean, I would say the real impact of that would be that you actually now can tell us, maybe not may not be able to sidestep doing intracranial EEG, but help direct where are other locations, which is a huge question.
Absolutely. I mean, guiding intracranial implantation in general is a huge field in of itself, right? Can we tell non-invasively kind of a better implantation mapping? I think that would be a great application of something like this.
Yeah, great job.
Thank you.
All right, Rachel, I'm going to jump in and reveal my ignorance about neuroengineering. So when you're making these models, in general, these are data-based models, right? You start with your data, and then you infer this model that connects 2 pieces of data that you have, in this case, the state at one time and the state at the next time. What's the potential role, or is there one for mechanistic models, for actually trying to represent some of the known physiology, either as a complementary way of doing these kinds of simulations, or at least potentially as a way of constraining your state model?
Yeah, that is an excellent question. And honestly, I wish that I knew a lot more about some of the more biophysical models that people use, because there are some really, really cool dynamics that come out of some of these neural oscillator models, and you can get these incredibly complex dynamics out of them that really match what's happening physiologically really well. It's almost the only reason that I feel like that I haven't tried to, well, it's more kind of like it's almost taking like two approaches to the same problem, I think. So I think that those mechanistic models are trying to say, can we find something about the underlying system that's causing this epilepsy in if we know that our model is indicating that this part of the thing is causing the dysfunction of this ion channel or blah, 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 right? Then you can say something about how we should treat and diagnose based on that, right? Whereas I think that this is going to be much more of a translational tool. So in the sense that this is going to be helping us determine kind of blind to some of the underpinnings of where these dynamics are actually coming from. If they can localize, then it almost like doesn't matter at that point, kind of what's underlying it, right? So it's almost, I think that I have been kind of treating it as kind of like two separate problems, but I think that I don't know how much I could incorporate in here, but I think that there is, that would be really powerful if you could have kind of a lot of these macro scale dynamics be modulated and kind of like supported by a lot of the more micro scale dynamics that you're going to get in a lot of these oscillator models.
Different version of the same question. What about connectivity mapping? Right.
Mm-hmm.
So if you think you know some about the connectivity from imaging, would you be able to overlay that on what you're doing here to potentially constrain or guide this?
Totally. People do that, and that's a really strong area of research right now that I think is super interesting. So if you use DTI data or something like that to inform some of the structural connectivity, and then you're building these dynamical network models to estimate the functional connectivity, and then you can kind of go from there. But also, and just in general, if you have a model of how the brain is functionally networked, then there's then this can kind of just be like kind of like the dynamics can go on top of that as well. So yeah, it's very and inherently this is just showing a functional connectivity network, right? But in the sense that this dynamical network is estimated in a way that it's kind of what I was showing here, where and I feel like I didn't explain this very well, but what the structure of the A matrix is actually trying to say is that at every element, it's going to be showing how the node that's in that specific column is directing the activity of the node in the row. So it's not symmetric on both sides. So it's not this static measure that you're just showing like some correlation between them. And I think that, and my whole PhD was all in these kind of more standardized computational neuroscience, like and connectivity metrics. But I think it's cool that you can actually say, okay, well, on this node is directing the activity of this node, but then down here, it's actually being influenced a lot by this node and whatever. So it's an interesting new way but still inherently a functional connectivity matrix of the system.
So can I ask that in a somewhat different way? I'm Dave Standard. I'm chair of the neurology department, and I'm not an epileptologist or an engineer. So maybe kind of an ignorant question, but you're modeling the connectivity of the sites you're recording from. But can you get to where you understand the intrinsic structure of the brain enough to model the sites that are not being recorded?
Yes, Dr. Fisher and I were actually talking about this earlier. Yes.
This is like, I think of it in genetics. We have directly sequenced genetics and we have imputed genetics. We know enough about chromatin structure that if we sequence certain parts, we'll understand what the rest looks like. Can you do the same thing here if you understand the intrinsic connectivity of the brain itself?
That's an excellent point. I've actually never really thought about it in that context. But yeah, Dr. Fisher and I were actually talking about this earlier, where I think that we only have data from what we can measure, right? So we have these intracranial electrodes implanted at certain locations that were clinically placed, and we don't have access to and even still, you're getting such a sparse sampling of the brain. So we're making these huge leaps. And at the end of the day, right, we're saying we're going to find the areas that are most resonant in the places that we measured, right? So that doesn't really have a whole lot to say about the places that we didn't measure, right? So in the sense of how kind of, yeah, like structural mapping or different things could really help. I think that, or any way of these non-invasive measures that can inform where we needed to implant to measure these things is a very, very good,
or maybe just accumulating enough data from individually implanted patients lets you build a model of the whole over time,
yes.
right? So if you had a large
yes.
number of these that collectively covered the entire brain, could you start to model the intrinsic connectivity and predict where the resonance would be, even though you don't have an electrode there?
Absolutely. This is something that, I was talking to another colleague about this, that it's kind of how like John Gottman and Birgit Fresher have done the high frequency oscillations across the whole healthy brain, right? So if you get a mapping of how many high frequency oscillations and what the HFO rate is going to be across the whole brain. Can we also do this with resonant frequencies, right? Can I say in this, and I think that we haven't explored at all about how resonance can be modulated in functional regions, right? And because they're hyperconnected, isn't any reason to think that there's not going to be some physiological resonance happening. So if there was a way that we could map across some of these healthier regions and kind of see if there is some base resonance that's supposed to happen, and then you can look for pathologically high resonance in certain areas, that kind of thing. It's a great idea. Absolutely.
So you showed at the beginning the oscillating bridge. Do you see the dynamical system, when you finally characterize it, as having individual nodes that become unstable while other ones maintain some stability, but the system is unstable? And if that,
go ahead. Yeah.
And I guess I'd ask you both questions and then you can kind of elaborate. And there's a follow-on question. If that's the case, is there some kind of analogy to the inverted pendulum problem where you oscillate the support point and there are ranges of oscillation frequency where you can actually stabilize the unstable situation?
I haven't encountered anything like the second one in my experience in this data so far. But to your first question, that's actually so funny that you asked that, because one of the other PhD students in our lab has been working on a localization problem. And still using dynamical networks, but what he was doing was called neural fragility, basically, so he would build his dynamical network from ictal EEG, so like building up to and then leading into a seizure. And he would look for, basically he would do these like small perturbations to see in each of the nodes, right, and all virtually, right? So you artificially input these small perturbations to kind of see what is the minimum level of perturbation you would need to destabilize the network, right? And so he said, if it only requires a little bitty input to destabilize the network, that node was more fragile than the rest of the nodes in the network, right? And he actually has incredible results showing that is incredibly localizing of seizure onset regions. His work was just published in Nature Neuroscience just the other day, which is pretty cool. So if you're interested in that, it's a great paper. And he showed how it was like similar to and like comparable or surpassed like all these other measures that people have used to localize seizure onset. So that's an excellent, very excellent thought. Our grad student just did it.
All right, thank you.
Yeah.
So a sort of follow-up to that is, have people thought about using this to control seizures? I mean, if you understand the system, right, you could inject current at, the right time in order to potentially control the seizure using a DBS or something like that.
Absolutely.
How would you, or not DBS, just the electrodes that you have there, you could keep them there and control the seizures. Is that the direction that people are thinking, or is there a reason that won't work?
No. So I think so basically one application that I think of this work that I haven't started to go in that direction yet, because I think almost like the more pertinent problem that I was going with is like, okay, well, if we can find these resonant regions, then we can know that we can stimulate a seizure from there, right? But then it obviously like logically follows, right, that maybe if we choose to not stimulate or choose to stimulate at specifically these off-peak frequencies, will that kind of deactivate this elevated network that's moving towards seizure, that kind of thing. Yeah, so that could be one way we could go with it. But yeah, I mean, I think that different stimulation, like basically finding stimulation paradigms to stop seizures, I think is going to be something that's incredibly, I mean, it's a very heavily sought out problem. But I think I think that it's going to take more of like, so you're going to have some of these like feedback mechanisms. So I think it's more of a controls problem at that point too. But I think that looking at these fact that there are frequency specific properties of the system, I think are really important, right? And obviously, right? That's like, so it's like if we can find and we can characterize these type of properties, then it can be really helpful.
And one kind of question that is a sort of a follow-up to this and sort of a follow-up to the question that Dr. Standard asked, is there a future direction for using anatomical or functional MRI or another tool to look at the anatomy of the whole brain to get a sense of what kinds of frequencies might be more common, or to make a model of what frequencies would be there? Is that a direction that people are thinking?
Yeah, so it's something that like I did get this question one time where people were like, well, have you thought about looking to see whether or not the resonant frequencies also correspond to the signature of the seizure itself, right? So like,
yeah, exactly.
Do the frequencies that you see in your resonance also translate to the actual ictal properties? And I think that's a very, very interesting open question that I would love to explore. And I think in a similar sense, you can get a lot of that from either scalp EEG, or you can get that from imaging. You might get that from MRI, right? If you can see like which like activation of different regions during different times, who knows? So yeah,
I was more thinking if you know the distance between the parts of the circuit that are resonating, then you can measure the speed of transmission and make an estimate. I don't know.
Yeah. I'm not sure. Yeah. That's an interesting thought for sure.
It's beautiful work.
Thank you.
Very, very neat. I've got one short question to go back. We've mentioned it before. The link between the state space model and your classic boring correlational functional connectivity network.
No, I love those. I know.
How tight is it if you take the state space model and try to generate or synthesize a functional connectivity matrix? Are they close, or are they completely unrelated?
I guess it would be in a sense of what kind of functional connectivity matrix you're working from. So in my PhD, I used a lot of more of like a cross correlation based functional connectivity. So if you're using, so I guess it'd be more of a, I guess, so the question of how
you could take the state space model and predict what the functional connectivity would be in terms of long-term correlation. Do they come out?
Yeah, I haven't looked at it directly, but I think that would be, I'm sure that there would be
the relationship was, or maybe there isn't, and they're looking at different things. I just was wondering.
Yeah, I'm not exactly sure. I think, well, and especially I would have to look. So specifically, this type of model is going to be building an effective connectivity matrix. So it's going to be directed. So I think that you'd have to look at kind of like a Granger causality measure or something like that, that specifically is a directionally based measure of connectivity.
Well, but you can always collapse because you're just destroying information. You could run the model with lots of random inputs and then see on average, what are the average correlations? I mean, you could try to synthesize it.
Sure. Yeah, I've not tried that before though. It's interesting.
Oh, neat talk though. Cool.
Thank you.
I have a question, and I also want to say a great work and really great talk. My question is about the experiment where you looked at the Bode plots and saw how they correlate with surgical outcomes and could distinguish with surgical outcomes. And when you first started showing that and you showed the Bode plots that look similar or different, my question was, how are you going to quantitate that? And then you showed the measure you developed to quantitate that, which was great. And there's clearly, by eye, again, looks like differences, but also there's some overlap. So there's other regions that aren't in the seizure onset zone that had similar magnitudes of your PW ratios. And so I guess what I'm wondering is, do you think that it would be possible to use these measures to really distinguish between what's in the seizure onset zone or not? Or is there other information that you would need together with this to make it so that they were non-overlapping and had more separation?
Yeah. So I think there are two parts to that question. So number one is that's actually one of the follow-up studies that I'd love to do before I leave my postdoc is trying to see what the actual prediction accuracy of saying of less of the surgical outcome prediction and being able to say, can I actually just classify seizure onset versus not just from the data? So that's something that I haven't actually done. But I think it's a hard problem. Number one, because we're working on clinical annotations that are generated by a ton of just disparate neural imaging data, right? And one thing that I think, so I don't know how well it would do, honestly. But more of, well, I kind of lost my train of thought. Sorry, hang on. Oh yeah. So no, I was going to say is that that it's something that I would never ask a clinician to only use my algorithm and not use any of their clinical expertise, right? So I think that I want this to be a decision support tool in a way that that hopefully, if we get it to a point where we can take it to a clinical trial and we can validate and we can say, yes, this helps you determine where the seizure onset is, right? But then if we have some node that has an incredibly high PW ratio, they're going to be like, I don't know about that guy, and be like, okay, yeah, throw it out. We don't need that, So I think that I definitely want this to be something that would never replace, right? So it's never going to replace their clinical expertise, I think, right? So yeah, and then the other thing that I was going to say was that, yeah, but along those lines, it's just so hard when there's not technically a ground truth, and especially true in surgical failures, right? So I think that those are so hard to evaluate in a lot of these studies because we know that there were some parts of the network that were still able to cause seizures that were left in surgical failures, but at the same time, it's just we have no idea what those regions really were. And if there's so many other different factors, what if they had forgotten to take their medication for so long and then the network looked different when they were in the hospital than other times. It's just, there's so many factors. So I think that in general, it's just a hard, yeah. So I totally feel you. And I think that
it's a hard problem.
It's a hard problem. Yeah, we're never going to have a perfect metric, but yeah. I just like to highlight reasons why, why my methods aren't perfect, So.
Thanks.
But yeah, it's a great question.